2N is diploid.
This?
Yeah.
Sure.
So I was gonna write it all down, and then I was like,
I kind of missed some stuff.
KDX.
I don't really know if it makes
Yay,
sense.
mitosis.
Oh god.
Nailed it. That is it.
So my hair is greasy this morning, so, and I don't own brush and blue, so I
I think I
and now
see these are remembering that.
Okay, so we discussed mitosis, and mitosis happens in body cells. And now we're going to talk about another type of cell division, basically meiosis, but this happens in sex cells, and it's involved in sexual reproduction. So it's very similar to mitosis in some aspects, but then it has a second round of division to cut that number in half. So meiosis reduces the number of chromosomes, because if a sperm cell had 46, and if an egg cell had 46, and they were to combine, you'd have 96 chromosomes, which means that you would not be human, okay? So the sex cells have to cut their number of chromosomes in half, so that way 23 in the egg, 23 in the sperm, they meet, you get 46. So Fleming, again, discovered chromosomes, and remember, he's the one that kind of coined the term mitosis, but it wasn't until Edward van Beneden noticed that there were different number of chromosomes in roundworms, and then they started to look at different species and noticed different numbers of chromosomes. So he noticed that there was a different number of chromosomes in gametes as well as the somatic cells. And he noticed that the gametes, the egg and sperm, only had 2 chromosomes, and in all the body cells of this roundworm, they had 4 chromosomes. And he was like, oh, what's going on here? And obviously somatic cells, somatic means body, means that they are non-reproductive cells. So mitosis is kind of responsible for like growth, development, repair. There's one more, I can't think of it. Growth, development, repair, and they'll come back to me. But those are the big 3 for mitosis. So cells in the body of sexually producing organisms can be, like I said, somatic cells, which are body cells, which means that they are diploid. They undergo the process mitosis to make more of them. Examples of somatic cells, anything that makes up your body that's not a sex cell. So muscle cells, nerve cells, stomach cells. Gametes, which are your sex cells, are haploid. They are produced through the process of meiosis. So if you are male, it's a sperm cell, and if you are female, it's an egg cell. Whoa. So back to Van Beneden, proposed that the egg and sperm fused in a single cell called the zygote. So a zygote is diploid. It is 2N. Okay, so N plus N is 2N. And all cells ultimately derive from this zygote. It was clear from the beginning that a mechanism is responsible for reducing the numbers in these sex cells, and that process is meiosis. So some life cycles, they have a haploid and diploid stage. So what I mean by that is, your gametes, they start off as haploid, but then when they fuse, they become diploid. And so this is sexual reproduction because you're combining genetic material from both sets of parents in the process of fusion to form that zygote. So life cycles of sexually reproducing organisms alternate between diploid and haploid variations. We'll talk more about this in second semester when we get to plants, but most algae spend most of their life cycle in a haploid state. And then when they release their spores and then they combine into a zygote, they undergo meiosis to cut their number in half, and then they just exist as haploid cells through the process of mitosis. Animals, however, the diploid state dominates. So zygote undergoes mitosis, producing all those diploid cells in you. Later on in life, some of those diploid cells go through meiosis, aka the sex cells, to form your haploid gametes. Okay. There are, however, some plants and algae that do alternate between multicellular haploid phases and multicellular diploid phases. So that's just the life cycle of some plants and algae. Like I said, second semester we'll dive more into that, especially when we talk about ferns and mosses, which stages plants are most dominant in. So, but animals, diploid state dominates. So here is an example with a fern, and we have a stage called a sporophyte, which means that it's diploid, and then it undergoes meiosis, producing spores, and these spores can undergo mitosis and produce a gametophyte, which is a haploid state of the organism, and it produces more gametes, and it fuses a gamete with another plant in the process of fertilization, form the zygote. Undergo mitosis, you get a sporophyte. So it alternates between diploid and haploid, different colors here. Now humans, we have sex cells, eggs are in the ovary, sperm in the testes. They undergo meiosis, and then when they fuse in the process of fertilization, we have a diploid zygote. All of our cells derive from that, but then there are a few cells, aka the cells in your sex organs, that go from diploid to haploid and then just produce haploid. So yes, your germline cells are set aside early in animal development to, again, give rise to your sex cells, and then everything else is diploid. Did Mr. Rourke ever torture you with the meiosis square dance? Oh, okay, just one.
I'm gonna ask him about it today.
Okay.
Put that out.
Not in this chapter, but later on after Thanksgiving break, we'll talk about how cells become specialized. But here are your gametes, the sperm and the egg. And when they fuse, you get a zygote forming an embryo. And then some cells, they start to differentiate. They start to form, all the types of cells inside your body. You have actually over 100 different types of cells in your body. And so we'll talk about that process of stem cells and how stem cells choose a pathway to become all the different types of cells inside of you. But some features of meiosis. It does vary in different organisms, mostly in how the chromosomes separate. But in general, diploid organisms go through 2 rounds of division. So we have meiosis 1, that's the first round of division where we have prophase 1, metaphase 1, telophase 1, anaphase 1. And then you go into meiosis 2, which is the second round, and then you hit prophase 2, metaphase 2, telophase 2, and anaphase 2. Another feature is that the homologous chromosomes pair in meiosis, and so we'll talk about this pairing. When they pair, they're actually forming a synapsis, and a unique thing called crossing over occurs. So here I have my homologous chromosomes, and they color-coded it. We can say this is from dad and this is from mom. We have 2 sister chromatids held together by a centromere. This is a tetrad or a bivalent. So they're paired up, forming a synapsis. And then what they do is they exchange genetic information. They literally swap genes through the process of crossing over. And this increases our genetic variation. And so that's why there's so many, combinations. I think it's like 23 to the 23. It's a really, really big number. You're really unique, is what I'm trying to say. But then after they exchange, you can kind of see they color-coded it. Oh, I got a little bit of mom chromosome, this is dad chromosome. And then you just have these, again, tetrad. And when they pull away, in the end of meiosis, if you're male, you'll form 4 sperm cells. But if you're female, only 1 of those cells will actually become the egg. They actually absorb all the other cells to form that egg. That's why egg cells are a lot larger than sperm cells. So zooming into synapsis. So after a synapsis occurs, what you actually have is a synaptical complex, which basically says that these 2 homologous chromosomes are stuck together, okay, forming this, there's like this protein in between that's keeping them together. It's holding them in place until crossing over occurs. This is the book definition. So the components of the synaptal complex, basically it's just a form of cohesion, just keeping them together, helps join homologous chromosomes so they pair up. It results in 4 chromatids, okay, there's 4 sister chromatids total, 2 homologous, closely associated. It can be described as a tetrad or a bivalent. So here's a diagram of synaptal complex, and you can see the kind of protein holding it together. And then what they'll do is they'll just swap genetic information. Real life pictures zooming in on the synaptal complex. It almost looks like protein threads, just kind of like thread sewing them together. So then the exchange of genetic material occurs, and we call this crossing over or genetic recombination. This happens in prophase 1. So prophase 1, homologous chromosomes pair up and cross over. Where they cross over are called chasmata. So here on a chromosome, they can cross over at multiple points. I know a lot of times in diagrams, they just show one crossover point. That's not the case. They can cross over many different points. So there could be several plasmata on homologous chromosomes. So then after prophase 1, we enter metaphase 1, and so the homologous chromosomes move to the middle of the cell. And then in anaphase 1, the homologous chromosomes are pulled apart from each other. And so the first division is termed reduction division, because this is where we actually start to cut the number of chromosomes in half. Now, I do want to stress that when we count chromosomes, we're not really counting chromosomes, we're counting centromeres. Okay, that's the thing that's holding the two X chromosomes together. And so here I have one chromosome, okay?
Instead of versus two, like of the actual, like since there's two connected, kind of, it's like it's just one. You're saying you're not counting the individual.
This is one.
Yeah, you're saying you're not counting like you count that, not like both of them as two.
Okay, I'm going to start over.
Well, I think I get what you're saying.
Okay, but I think I missed a step. I wanted to talk about something else. Okay, let's say that we have the egg and the sperm fuse, and we'll say green is dad and mom is red, okay? And these are going to become sex cells. So then the first step is that the chromosome, so this is a chromosome and this is a chromosome. I should draw the centromeres. Even linear chromosomes have centromeres. Okay, so the first step in prophase 1 is that they replicate. Okay. And then they pair up, forming the homologous chromosome. And then they line up in the middle during
Now
metaphase.
there's only 1 century.
There's still 1 centromere here and 1 centromere here. Okay, so
You're welcome.
here I have a chromosome, right? And here I have a chromosome. So I have 2 chromosomes, but I have how many sister chromatids?
4.
4 sister chromatids. Okay, so then we go through meiosis 1, and these guys will get pulled apart. Okay, so over here, they're separated, so sad. Okay, and then they'll go through meiosis 2, and now the homologous chromosome will be pulled apart into sister chromatids. So now I have 1 chromosome, 2 sister chromatids, right? So then after meiosis, I get something like this, which means I end up with 4 cells. This is 1 cell.
And they're not actually just green and red, like they're intermingled.
Yeah, there's crossing over. There's little bits of each other, but I didn't want to go through that step. So I end up with 4 cells, and 4 sister chromatids. I like keeping track of sister chromatids. It just makes more sense because the chromosome definition is weird. Chromosomes, you're basically just counting centromeres. All right, so here's metaphase 1. Homologous chromosomes pair up. Right down the middle, you can see that crossing over has occurred on some. And then anaphase 1, they get pulled away from each other. So our homologous chromosomes separate, they're no longer homologous, and now we just have 2 sister chromatids for this chromosome. Now remember, meiosis has 2 rounds of division. And so the first round, you have DNA replication. I drew linear and then I drew partner, okay? But in meiosis 2, there's no DNA replication. And this is how the number gets cut in half. So we call that first meiotic division reduction. And the second meiotic division, meiosis 2, is very similar to mitosis, except there's no DNA replication. So if you understand mitosis very well, you know exactly what's going to happen in meiosis 2. It's just meiosis 1 that's different because you get that DNA replication, the pairing up of the homologous chromosomes, the crossing over. How they line up on the metaphase plate is a little bit different. They straddle the line, while in mitosis they were right on the line.
Meiosis 2 is like the same as mitosis.
Mm-hmm. So here's a diagram of meiosis 1 with our homologous chromosomes pairing up and they're swapping genetic information. And notice how they line up on the metaphase plate. They straddle the line. Homologous chromosomes get pulled apart. We get 2 cells. You can see the crossing over has occurred. So we have 2 chromosomes here, 2 chromosomes here. It goes through cytokinesis and then it moves on to prophase 2. where there's no DNA replication, okay, they line up on the metaphase plate directly, just like in metaphase of mitosis. The chromosome gets pulled apart into its sister chromatids. You get telophase for, well, new nuclear envelopes form cytokinesis, and so now you have 4 haploid cells. And like I said, in males, all of these will become sperm. In females, one of these will become the egg, and it will absorb all the other haploid cells.
So you said that in prophase 2, there's no new genetic information?
There's no DNA replication,
Yeah,
and there's
so
no crossing over.
So the telophase is it just splits up when it already is in there?
Yep.
Telophase 2.
So now I'm just going to go into the nitty-gritty details that I've just said. So prophase 1 sets the stage for that reductive division. There's interphase beforehand, but then after interphase, we go into meiosis 1 where DNA starts to condense. Our homologous chromosomes form a synapse to get ready to cross over. The nuclear envelope breaks down, spindle fibers form, attach to the kinetochores, and start moving them a little bit. We have that synapsis. Yeah, this is just review. Your book gives you an overview and then it goes into nitty-gritty details. So yes, we have all 4 chromatids in close association. They will cross over at some points. What do we call the crossover points?
Not the centromere.
No, starts with the letter C. Starts with the letter C.
The chiasmata.
Yeah, the chiasmata. Yep, very good. So we have crossing over. We also can call it genetic recombination. They exchange. When crossing over is complete, that synaptimal complex breaks down. They're still attached at the chiasmata, but at other areas they start to fall away from each other. The 4 chromatids do not separate completely. They're still held together by their centromeres and by the chiasmata. There's prophase 1, and they can actually break down prophase 1 into several more steps, but you don't need to know that. It's just showing you, oh, hey, synaptical complex forms. Then it's all along the line. Then they cross over, and now we're done. Okay, during metaphase, they are aligned. They straddle the metaphase plate. It's random how they get lined up. Also, the microtubules, they attach to the kinetochores of the homologs, not to the sister chromatids. So I think you see that in the diagram here. So here's a possibility of how they can line up. You can line up like all the blues on one side and all the reds, and you get this. Or maybe it's blue-red and then red-blue, and then they separate. And this is just like one of the many different combinations. I mean, remember, you have 23 chromosomes, or 23 pairs, I should say, sorry. 46, okay? And so many different combinations of how they can line up. So you get into the anaphase. The homologous chromosomes are pulled away from each other, so now you're just left with individual chromosomes. The sister chromatids are still attached by the centromere. Again, due to random orientation, a pole may receive either maternal or paternal homologs. We call this independent assortment. Keep this vocab word in mind when we get to Gregor Mendel, the Austrian monk, because we'll talk about independent assortment again. So then after metaphase 1, we have anaphase 1. Homologous chromosomes move to opposite ends of the cell. And then telophase completes meiosis 1, where you get that new nuclear membrane around each of the daughter cells. The sister chromatids are no longer identical due to crossing over. That's another unique thing. So then in meiosis 2, it's just like mitosis without the DNA replication. Nuclear envelope breaks down. Spindle fibers form and start to connect to the kinetochores of the sister chromatids. We get into that metaphase 2, where they line up on the line. Okay, this is showing metaphase 1, they're comparing it. So now the chromosomes will be pulled apart into sister chromatids. So there's anaphase, sister chromatids. Yes. Sure. ahead. Okay. Into anaphase 2, and then finally reaching that telophase where you get 4 haploid daughter cells.
So would you get like 8 totals instead of 4 in the first half?
No, you only get 2 in the first half, and then the 2 become 4.
Oh.
So it starts off like this.
Oh,
And that's why we say 4. And this
so 4 at
is,
the end of all of it.
yeah, this is meiosis 1 and this is meiosis 2.
So then cytokinesis doesn't happen.
Yes, it does.
It
Yep.
happens in both of them?
Yes. Yes. Because that's how you divide the cytoplasm. So eventually it will pinch.
So that's all cytokinesis does, is completely close them apart.
Yep. So the final result of meiosis, you get 4 haploid sets of chromosomes. These will turn into gametes in animals, or if they're plants, fungi, and protists, they can become multicellular haploid organisms. They will just divide mitotically. Now, there can be some errors in the process of meiosis, but we probably should address them. So, one, meiosis needs to be accurate to make sure you get the correct number of chromosomes. You want to make sure that we pull them away equally so that all the cells get the correct number of chromosomes. However, sometimes it doesn't happen, and we call that non-disjunction, where one gamete lacks a chromosome and one has 2 copies. And so this is what happens when you get trisomies and eukaryotypes with, like 3 copies of chromosome 21. You saw that. You get Down syndrome. 3 copies of chromosome 18, Edwards syndrome. If you get 3 copies, like 2 X's and a Y, you could be, I think it's Klinefelters, right?
Is it possible?
Or is it Turner's? I forget.
Is it possible to have like 48 chromosomes? Then you have 47.
Yeah, you could be XXXY. That's the only case.
What would that? Is that trisomy?
XXXY.
Oh.
What? Excellent, thank you.
So what would that be if you had what you just said?
No, they have like weird names for it. I remember reading a bunch of scientific articles in college. Let's see what it says.
But could you have like trisomy 21 and 18?
Yeah, you could. It's very, 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 very rare, but it's a possibility. They call it. They literally just call it XXXY syndrome, and sometimes they just call it Klinefelters, but it's just more severe.
Like what would
Maybe we can
happen?
call it Einklein.
Yeah, what would this one be like?
Well, I guess I can just click on it. This is from Wikipedia, so you can't.
Close.
Wide variety of symptoms associated with it, including cognitive and behavioral problems, taurodontism, and infertility.
I think this is what happens when they're all blacked out. I found a picture.
Wow.
I don't think that's what happens.
Okay.
Matt, I think that's.
Okay, so now a lot of times if the gamete has an incorrect number of chromosomes in the haploid cell, the egg or the sperm, or like after the fusion of egg and sperm, a lot of times like your body will just abort. I should say not everyone's body. Female bodies will abort the embryo because it knows it's not
So
right.
is that like a miscarriage or is that not even?
No, yeah, it would be considered.
So if someone has a miscarriage, it's not all it like could be.
It could be because the body did it. Because it's like, okay, something's not right. Or like, maybe there wasn't enough nutrition, or the placenta wasn't ready. Like, just got the wrong messages, the wrong hormones. So. Okay, so sum it up. Know the differences between meiosis and mitosis. I guarantee you on your exam, you have to at least know 4 differences between meiosis and mitosis.
Meiosis winds up on the border, like it's not right in the center. It's on the
They straddle
Straddle,
the
yeah.
metaphase plate. But you got to know that homologous chromosomes straddle the metaphase plate.
Okay. Not just meiosis.
What are some other differences? Meiosis is for like gametes.
Yes, it's for sex cells. Happens in sex cells. What's another difference?
Meiosis means 4 at the end.
4 4 cells versus 2 cells.
4 haploid.
Okay, what's another thing that happens in meiosis that doesn't?
Meiosis goes through peanut twice.
Yeah, 2 rounds of division.
Is that kind of the same as 4? Or no?
We like to say 2 rounds of division.
Or homologous strand.
Any others?
Something.
There's a few more.
Wow.
There's actually like 6 to 8.
Different.
What about prophase? What happens in prophase in meiosis?
New genetic
Or
material.
DNA replication.
Or DNA
there's no
replication.
DNA replication.
I thought it
In the
was
second round, but then what happens also in prophase?
They cross over.
You cross over. That doesn't happen in mitosis.
Oh.
Okay, so yes, there are at least 4 distinct features, but there's many more if you think about it. The first difference in meiosis, you get the pairing of homologs, and then that crossing over between maternal and paternal homologous chromosomes. This one is talking about how the chromosomes move. So, like, yeah, basically in anaphase of meiosis, homologous chromosomes separate from each other. The attachment of the sister kinetochores to the same pole in meiosis 1. So in this mitosis and meiosis, they connect to the kinetochores of both sister chromatids, but in meiosis, it's to the kinetochores of the homologous chromosomes, the maternal and paternal. Suppression of replication in prophase 2 is another distinct feature. So here's a comparison picture between mitosis and meiosis. We have prophase, the chromosomes duplicate, they line up on the middle, the chromosome becomes sister chromatids, and you end up with diploid cells. In meiosis, homologous chromosomes from maternal and paternal pair up, you get that crossing over, the homologous chromosomes line up on the metaphase plate, but they straddle it, okay? After the first round of division, just homologous chromosomes, they separate, so now you just have your 2 sister chromatids to make up that chromosome. They go through another round of division, very similar to mitosis, and you end up with 4 haploid cells that are genetically different from each other. These guys are identical. These are genetically different.
So that's why it's like one sperm fertilizes the egg versus a different sperm fertilizing the egg. You get a different. Radical.
So a table, the last slide in this, again, just compares like DNA replication, when does it happen, how many rounds of division, does synapsis occur, number of daughter cells at the very end, and what's the role? Growth, repair. In meiosis, it's for sexual reproduction. So I'm going to torture you, I think, with the meiosis square dance.
Yes.
Something that I had to watch. So I think I'm just going to pass on the suffering. Whoops. If you go to Gustavus, the second day, they have a square dance for the freshmen. Supposedly, the person you dance with will like turn out like you guys will date and possibly get married.
Did that happen to you?
No, I didn't go to the square dance.
Oh.
Hannah, I said next school dance. I'm gonna go up to someone and say hi. Do you want to buy this?
I'm